Welcome to Bedtime Science. Did you know that a man and a woman lifting the same weight will trigger almost identical growth signals inside their muscle cells, even though their hormone levels are vastly different? Tonight we look at the quiet chemical messengers, hormones, that silently shape male and female bodies from before birth to old age. In the next moments, we will gently discuss how testosterone and estrogen build different baselines during puberty while leaving the underlying cellular machinery remarkably similar. How the monthly rhythm of estrogen and progesterone reaches into the brain to adjust the sensitivity of mood and reward circuits. What happens when hormones decline in later life, and why bone loss follows two very different timelines in men and women, and how the stress hormone cortisol responds differently in male and female bodies placed under identical social pressure. Before we begin, if you find comfort here tonight, consider subscribing to join our quiet community. And if you are still awake, you can leave a soft comment about what topic we should explore next. Now let's begin. Right now, inside your body, molecules smaller than a grain of dust are slowly deciding how your bones hold together, how your muscles respond to effort, and how your brain feels about drifting off tonight. These molecules are hormones. They don't work like nerves. A nerve signal is fast. It can travel along a single fiber at 100 meters per second, delivering a command from your brain to a fingertip in a fraction of a moment. You touch a hot pan. The message races up your arm. Your hand pulls back. Hormones operate on an entirely different schedule. They're released from glands into the bloodstream and they drift. They circulate through your entire body, sometimes for minutes, until they find a cell that's ready to receive them. Think of nerve signals as phone calls, direct and instant. Hormones are more like letters carried by a slow current, floating until they reach the correct address. And this slowness isn't a design flaw, it's the whole point. Hormones carry instructions that aren't meant to be acted on in milliseconds. They carry instructions meant to shape things over hours, over days, over months and years and decades. They tell your bones how quickly to rebuild themselves. They adjust how deeply your brain responds to a reward. They set the pace at which your body stores energy or burns through it. The place where most of these instructions originate is a structure no larger than an almond, sitting near the center of your brain. It's called the hypothalamus. This tiny cluster of neurons acts as a bridge between your nervous system and your endocrine system, which is the network of glands that produces hormones. Your hypothalamus reads signals from the blood. It reads signals from the brain itself. It registers light entering your eyes, temperature on your skin, glucose concentration, stress levels. It gathers all of this and translates the information into chemical commands. Hanging just below the hypothalamus is the pituitary gland. It's roughly the size of a pea, maybe a bit smaller. And from this pea-sized gland, hormones travel outward to the thyroid, to the adrenal glands perched on top of your kidneys, to the ovaries, to the testes, instructing each one to release its own specific chemical messengers into the bloodstream. Two structures, both tiny. Together, they direct the entire hormonal activity of your body. Here is something that surprises most people when they hear it for the first time. Male and female bodies share the same hormones. Testosterone is not exclusively male. Estrogen is not exclusively female. Both exist in every human body. The difference lies in the ratios, in the concentrations. A typical adult male produces roughly 10 to 20 times more testosterone than a typical adult female. But a woman's body still produces testosterone in the ovaries and the adrenal glands and uses it for bone upkeep, muscle repair, and certain aspects of mood regulation. A typical adult female produces more estradiol, which is the most active form of estrogen. But a man's body also produces estradiol, converting some of its testosterone through an enzyme called aromatase. And this estrogen is essential for male bone health and brain function. Same molecules, different concentrations. From those different concentrations, two somewhat different physiological outcomes emerge across a lifetime. But here's what matters even more than the concentration of a hormone circulating in your blood. It's how loudly the cells are listening. This is called receptor sensitivity. A hormone molecule drifting through the bloodstream does nothing on its own, nothing at all. It has to find a receptor, a protein on or inside a cell that recognizes its specific shape, its specific chemical signature. When the hormone binds to that receptor, the cell changes its behavior. It might begin producing a new protein. It might stop dividing. It might release its own signal downstream, triggering a chain of events in neighboring tissue. The number of receptors a cell carries matters. How tightly those receptors grip the hormone matters. Two people can have identical testosterone levels in their blood, but if one person's cells carry more androgen receptors, or if those receptors bind testosterone with greater affinity, the downstream effects will differ. Your cells listen at different volumes. And that's why simply measuring a hormone level in a blood test gives you only part of the story. The full picture includes how the signal is received at the cell's surface, how it's amplified inside the cell, and how the cell translates that molecular conversation into a physical change you'd actually notice. This is the shared blueprint, same hypothalamus, same pituitary, same hormones, same receptor families, a system built on common architecture tuned to different settings. And the moment when that tuning diverges most dramatically is puberty. For the first decade of life, the hormonal profiles of boys and girls are not dramatically different. Both produce small amounts of testosterone. Both produce small amounts of estrogen. The machinery is present, but it's running quietly like an engine at idle. Not much output, just ticking over, waiting. Then something shifts. Deep in the hypothalamus, a gene called KISS1 starts to change. This gene encodes a protein called kisspeptin, and kisspeptin acts as the molecular trigger for the entire cascade of puberty. Before puberty, the KISS1 gene is largely silenced. It's held quiet by a chemical tag called a methyl group, a small molecule attached to the DNA near the gene's promoter region. This is an epigenetic mechanism. Your DNA sequence itself hasn't changed. Not a single letter of the genetic code is different. But the methyl group prevents the gene from being read, like a bookmark placed over a paragraph in a book, telling the cell to skip this part. When that methyl group is removed, a process called demethylation, the KISS1 gene becomes active. Kisspeptin floods out, and kisspeptin signals the hypothalamus to begin releasing GnRH, gonadotropin-releasing hormone, in pulsing waves. Those pulses travel the short distance to the pituitary. The pituitary responds by releasing two hormones of its own, luteinizing hormone and follicle-stimulating hormone. These travel through the bloodstream to the gonads, instructing them to begin producing sex hormones at adult concentrations. In boys, this means a surge of testosterone. In girls, a rise in estradiol and the beginning of the monthly ovarian cycle. The testosterone surge in boys coincides with a rapid increase in muscle mass. Here's the thing. This is when the baseline difference in muscularity between men and women gets established. Not during training, not during adulthood, during puberty. The testosterone of male puberty sets a higher starting point for total lean mass, building more muscle fiber, more cross-sectional area, and this head start persists throughout life even if testosterone levels later fluctuate within their normal range. But the muscle cells themselves don't become different. We'll come back to that. It's one of the quieter findings in exercise physiology, and it reshapes how you think about strength, training, and the role of testosterone in physical performance. There's another dimension to puberty that goes beyond the physical. Dr. Catherine Page Harden, a psychologist at the University of Texas, has described the mismatch that puberty creates between the body and the brain. She observed that during puberty, boys seem to change overnight. Their height, their musculature, their hormonal drive, all accelerating rapidly. But their cognition, as she put it, was not necessarily maturing as quickly as their height or their musculature or their hormones. The prefrontal cortex, the part of the brain responsible for impulse control, long-term planning, and risk assessment, doesn't finish developing until approximately age 25. Some recent Cambridge research suggests the strongest structural turning point occurs around age 32. Puberty begins years earlier, between ages 8 and 13 in girls, between 9 and 14 in boys. So there's a window, sometimes lasting more than a decade, where the body has reached a level of hormonal maturity that the brain's decision-making architecture isn't yet equipped to manage. The engine is at full power. The steering is still being built. A study using the ABCD cohort, tracking roughly 11,500 children aged 9 to 13, confirmed that earlier pubertal timing was associated with accelerated brain development, particularly in subcortical and frontal regions in females and subcortical regions in males. But here's what stood out. Physical measures of puberty had stronger associations with brain maturity than hormonal measures did, suggesting that psychosocial mechanisms, the way the world treats you differently when your body looks older, may carry more influence than the direct hormonal effects on neural tissue. Think about that for a moment. It's not just the hormones acting on the brain from the inside. It's also the social world reacting to the body from the outside. Both push development forward. Both shape who you become. And the changes happening during puberty reach further than hormones alone. They reach into the genome itself. DNA methylation, the same mechanism that silenced the KISS1 gene before puberty, shifts across thousands of sites throughout the pubertal transition. These methylation changes alter which genes are more active and which are less active without touching the underlying DNA sequence. A methyl group is small, smaller than a molecule of water, but placed on a specific cytosine residue in the genome, it can change how a gene is expressed for years, for decades, possibly for the rest of a person's life. A study published in BMC Genomics found that methylation sites differing by biological sex were largely independent of sites undergoing change across the timeline of puberty. Both sets of methylation networks, the sex-related ones and the time-related ones, reflected salivary testosterone levels and were enriched for androgen response elements. The hormonal environment of puberty was literally reprogramming the epigenome, writing new instructions onto the same DNA. In female mice, inhibiting DNA methylation decreased estrogen receptor alpha expression in the arcuate nucleus of the hypothalamus. In males, inhibiting demethylation enhanced it. Testosterone was programming receptor levels through epigenetic pathways, not just acting on existing receptors, but determining how many receptors future cells would carry. This is the long shadow of puberty. It doesn't just change how you look at 14. It changes how your genome reads its own instructions for the rest of your life. Research from Dr. Hardin's group has established that DNA methylation measures of biological aging can be quantified in children decades before aging-related disease shows up. In 8 to 18-year-olds from the Texas Twin Project, girls who had experienced their first menstrual period showed a somewhat faster pace of biological aging compared with same-aged girls who had not yet reached that milestone. Children who were more advanced in pubertal development showed higher markers of chronic inflammation, even after the researchers controlled for socioeconomic disadvantage. Earlier puberty, faster biological aging, a connection written in methyl groups on the surface of DNA. This aligns with something called the epigenetic clock theory of aging. The idea is that the same developmental program optimized to produce a reproductively mature adult also sets the timer for the gradual decline that follows maturity. The very mechanisms that build the body during puberty may, in a quiet way, begin the process of wearing it down. And all of it starts with a methyl group being removed from a gene in the hypothalamus. One small chemical tag, lifted from one stretch of DNA in one tiny brain structure. And from that single molecular event, the entire trajectory of a human body changes. The night is still early. Let your mind rest with this idea for a moment before we continue. Let's stay with the female body for a while and follow what happens after puberty establishes the monthly hormonal rhythm. Every month, roughly every 28 days, though the range varies from person to person, the female body cycles through a sequence of hormonal shifts. Estradiol rises during the first half, the follicular phase, reaching a peak just before ovulation. Then progesterone rises in the second half, the luteal phase, while estradiol dips and then partially recovers. This is often described as a reproductive cycle, and it is, but its effects reach far beyond the reproductive organs, far beyond. Estradiol and progesterone are lipophilic molecules. They dissolve easily in fat. Because of this, they cross the blood-brain barrier with little resistance. They enter the brain directly, moving through the membranes of cells as easily as water soaking into cloth. The highest concentration of progesterone anywhere in the brain is found in the amygdala, a structure involved in processing emotion, particularly responses to threat and social signals. Estrogen receptors, both the alpha and beta types, are expressed throughout the amygdala, the hippocampus, the hypothalamus, and the prefrontal cortex. These hormones aren't just passing through the brain. They're adjusting its chemistry month after month. Estrogen, broadly, enhances the activity of glutamate, serotonin, and dopamine, which are excitatory and mood-related neurotransmitters, while it suppresses GABA, the brain's primary inhibitory signal. Progesterone does something close to the opposite. It suppresses glutamate. It enhances GABA, and it has variable effects on serotonin and dopamine depending on the brain region and on prior estrogen exposure. So across the course of a single month, the female brain shifts between two different neurochemical environments, Not dramatically, not dangerously, but measurably. Let's look at serotonin first. There's a gene called TPH2. It encodes an enzyme, tryptophan hydroxylase 2, that serves as the rate-limiting step in serotonin production within the brain. This enzyme operates in the dorsal raphe nucleus, a cluster of neurons in the brainstem that supplies serotonin to much of the rest of the brain. If you think of serotonin as the brain's mood-stabilizing molecule, then TPH2 is the bottleneck in its production line. However much TPH2 you produce determines the upper limit of how much serotonin you can make. Estradiol directly increases TPH2 expression. It does this through estrogen receptor beta. Researchers here and Handa identified a specific estrogen response element located at -700 and 92 nucleotides upstream of the TPH2 transcription start site. When serotonergic cells were treated with estradiol, TPH2 promoter activity increased in a dose-dependent manner, with the maximum effect showing up at 24 hours. When the researchers deleted or mutated that response element, the effect disappeared. When they blocked the estrogen receptor with an antagonist, the effect disappeared. Clear, direct, confirmed at the molecular level. In plain terms, when estradiol is high, the brain produces more of the enzyme it needs to make serotonin. When estradiol falls, that production slows. But estrogen doesn't just boost serotonin production. It also slows serotonin removal. The serotonin transporter, known as SCRT, is responsible for pulling serotonin back out of the synaptic cleft, the tiny gap between two neurons where signaling happens. Estrogen inhibits SCRT, meaning serotonin stays in the gap longer, amplifying its signal. Estrogen also inhibits monoamine oxidase, an enzyme that degrades serotonin after it's been used. More production, slower removal, slower degradation. The combined effect, higher serotonin availability in the brain's limbic and cortical circuits during high estrogen phases of the cycle. Now consider what happens in the late luteal phase, the days before menstruation, when both estradiol and progesterone are withdrawing. Estrogen's serotonin-enhancing effects fade. That's one shift, but progesterone's withdrawal adds another layer. During the luteal phase, progesterone is converted into a metabolite called allopregnanolone. This metabolite is a potent enhancer of GABAA receptor activity, meaning it boosts the brain's main inhibitory, calming system. Allopregnanolone concentrations reach approximately 7 nanomoles per liter during the luteal phase. When the luteal phase ends, allopregnanolone drops to around 1.5 nanomoles per liter. That's a steep withdrawal of calming neurochemical activity, happening at the same time that serotonin support is declining. The result is a temporary shift in the brain's neurochemical balance. For most women, this shift is subtle. A slight change in mood, a bit less emotional buoyancy. In some women, particularly those with premenstrual dysphoric disorder, PMDD, the shift produces pronounced disruption. Research has confirmed that premenstrual symptoms vanish when ovarian hormones are suppressed using GnRH agonists, and the symptoms come back when progesterone is added back. The exposure to progesterone during the luteal phase and its subsequent withdrawal is the direct trigger. Now let's follow the dopamine side. Estrogen increases dopamine synthesis. It decreases dopamine degradation and reuptake. It upregulates dopaminergic receptors, particularly in the prefrontal cortex, a region with unusually high estrogen receptor density compared to other cortical areas. When estrogen is elevated, the prefrontal cortex runs on a slightly richer dopamine supply. The question of whether D2 receptor availability changes across the menstrual cycle has produced mixed results. A study in cynomolgus monkeys found D2 receptor availability was about 12% higher in certain brain regions during the luteal phase compared to the follicular phase. But a careful 2021 human study, scanning 16 women during 2 menstrual phases using PET imaging with a radio tracer called folapride, found no significant change in D2 receptor availability in the striatum across the cycle. The researchers concluded that circulating estrogen levels do not affect dopamine D2 receptor availability in the human striatum. So the picture is complicated. Estrogen clearly modulates dopamine systems in the prefrontal cortex, but in the striatum, the deep reward hub, the effect on dopamine receptors may be more subtle than animal studies first suggested. In the prefrontal cortex, allopregnanolone, the progesterone metabolite we talked about, inhibits dopamine-induced glutamate release. This is one mechanism connecting progesterone levels to shifts in cognition and executive function during the luteal phase. The brain regions where these hormonal effects are most pronounced, the amygdala, the nucleus accumbens, the prefrontal cortex, are the regions governing mood, reward evaluation, and decision-making. These aren't peripheral structures. They're the core architecture of how your brain evaluates what matters and what to do about it. And here is the quiet insight that ties this section together. None of this is dysfunction. The monthly neurochemical shift isn't a malfunction. It's not a vulnerability. It is the body cycling through different metabolic and neurological states as part of a reproductive rhythm that evolved over hundreds of thousands of years. The brain's chemistry isn't static in either sex, but in women it follows a visible, measurable oscillation that scientists have now mapped down to the level of individual gene promoters and receptor subtypes. The brain adjusts, month after month, a slow tide of chemistry that most people never consciously notice, running beneath the surface of daily life like groundwater flowing under a quiet field. Let's turn now to the question of muscle and strength, because this is where one of the most persistent myths about hormones starts to come apart under careful examination. The common belief goes something like this: testosterone builds muscle. Men have more testosterone, so men build muscle more easily, and the cellular processes of muscle growth must work differently, or at least more powerfully, in male tissue than in female tissue. Dr. Lauren Colenso-Semple earned her PhD from McMaster University under the supervision of Professor Stuart Phillips, one of the leading authorities in muscle physiology anywhere in the world. She spent years testing this assumption with the most rigorous experimental methods available. Her conclusion is direct. When we look at how the muscle responds, specifically muscle protein synthesis in response to exercise or nutrition, there are no differences. No differences. Let that settle. A foundational 2009 study published in the Journal of Applied Physiology measured muscle protein synthesis rates in 16 healthy adults. 8 men, 8 women, matched for age and body mass index. The researchers used stable isotope-labeled amino acid tracers, a technique that lets you track exactly how fast muscle cells are assembling new protein, molecule by molecule. The basal rate of muscle protein synthesis wasn't different between men and women. The statistical test returned a p-value of 0.49. That's about as far from a meaningful difference as you can get. When both groups were given amino acids and insulin to stimulate muscle growth, protein synthesis increased by approximately 50% in both sexes. The size of that increase was nearly identical: 0.019% per hour in one group, 0.018% per hour in the other. P-value 0.93. Indistinguishable. The researchers also measured the key signaling proteins inside the muscle cells: ACT, mTOR, P70S6K. These are the molecules that form the cascade connecting a training stimulus to actual protein construction, the internal wiring that turns effort into growth. No differences between sexes in any of these proteins. So if the cellular machinery works the same way, where does the visible difference in muscularity come from? Puberty. During the testosterone surge of male puberty, boys accumulate more total muscle mass, more fibers, more cross-sectional area. This is a quantitative head start, but it's not a qualitative advantage. The machinery inside each muscle cell works identically. Here's another finding that challenges what most people assume. Within the normal male testosterone range, roughly 300 to 900 nanograms per deciliter, having a higher level doesn't predict a better training response. A man at 400 doesn't build meaningfully less muscle than a man at 600, assuming both train and eat in similar ways. The same principle holds for women within their normal testosterone range. And the acute spike of testosterone that occurs right after a hard workout, the post-exercise surge that some trainers have built entire training philosophies around, it doesn't predict long-term muscle adaptation. It's a transient hormonal blip, not a growth signal. Only at supraphysiological levels, the kind produced by exogenous anabolic steroids that push testosterone far beyond what any human body generates naturally, do dramatic differences in muscle growth emerge. That's the real game changer, as Dr. Colenso Semple has described it. Not the natural variation within normal ranges, but the pharmacological amplification to concentrations the body was never designed to sustain. There's also a technical wrinkle worth mentioning. Testosterone blood tests, the assays clinicians use, have historically been calibrated for the male range. At the lower concentrations typical in women, these assays lose accuracy, which means we actually know less about female testosterone physiology than most people assume. The measuring instruments weren't built to capture it precisely. What about muscle fiber types? In people who don't train, the distribution of type I fibers, slow-twitch, endurance-oriented, and type II fibers, fast-twitch, power-oriented, is approximately 50-50. The sex difference in this distribution is maybe 1 percentage point in either direction, practically negligible. And fiber type is adaptive. It shifts with training. Endurance work pushes fibers toward type I characteristics. Strength work pushes them toward type II. These adaptations can be quite dramatic in both sexes, as Dr. Colenso Semple has noted. Both men and women carry the potential for large shifts in either direction. The idea that women need entirely different training programs from men, different rep ranges, different exercises, different periodization, doesn't hold up. It's not supported by the data. Men and women respond to exercise in the same way at the cellular level. The stimulus that triggers muscle growth in a man triggers it through the same pathways in a woman. Dr. Colenso Semple's own 2024 randomized controlled trial, published in the Journal of Physiology, tested whether fluctuations in ovarian hormones across the menstrual cycle influence muscle protein synthesis. 12 participants completed two 6-day study phases in randomized order, one during the late follicular phase when estrogen peaks, and one during the mid-luteal phase when progesterone is highest. In each phase, they performed unilateral resistance exercise, training one leg. The results were unequivocal. Exercise stimulated muscle protein synthesis, but there was no effect of menstrual cycle phase on that synthesis. No interaction between exercise and cycle phase. No effect on myofibrillar protein breakdown either, with a p-value of 0.24. Metabolomic analysis confirmed no notable metabolic pathway differences between the two phases. The muscle doesn't care where you are in your cycle. It responds to effort the same way, with the same molecular signals at the same rate. This finding sits quietly in the scientific literature. It doesn't generate dramatic headlines, but it slowly, steadily reshapes how researchers understand the relationship between hormones and physical performance. The cells receive the same instructions. They respond identically. They just start from different baselines, set years or decades earlier during a pubertal window that lasted only a few years. Let's shift to a different kind of hormonal response now. One that doesn't unfold over months of training, but in the space of a few minutes under social pressure. The Trier social stress test, developed in Germany, is the gold standard laboratory method for studying how the human body reacts to psychological stress. The protocol is straightforward. You're brought into a room and asked to give a public speech, usually a mock job interview in front of evaluators who sit stone-faced and give you nothing. No smiles, no nods, no encouragement. After the speech, you perform mental arithmetic aloud. You subtract a number like 13 from a 4-digit starting number, over and over again, while the evaluators watch and correct your mistakes. It's uncomfortable, deliberately so. And the body responds. The hypothalamus kicks the HPA axis into gear. CRH is released. The pituitary answers with ACTH. The adrenal glands, perched on top of the kidneys, pour out cortisol. A 2022 meta-analysis pooling 76 separate study samples confirmed what individual studies had been showing for years. Males produce a higher cortisol response to the TSSD than females. The specific numbers: baseline salivary cortisol averaged 8.61 nanomoles per liter in men and 7.08 in women. A modest gap, but peak cortisol after the stress test averaged 17.15 in men and only 10.87 in women. That's approximately 58% higher peak cortisol in men than in women. Same room, same task, same social pressure, very different hormonal output. But here's the part that complicates things. Men and women report similar levels of subjective stress. They feel about the same amount of anxiety, the same discomfort, the same sense of being evaluated. It's the cortisol, the physiological marker, that diverges. Only about 25% of women showed a cortisol response to both a standard and an alternative stress task, compared to 90% of men. The female body isn't responding less. It's responding through different channels. A study by researchers Childs, D. Lugos, and DeWitt compared TSSD responses across 3 groups: men, women in the follicular phase of their menstrual cycle, and women in the luteal phase. Only men showed a statistically meaningful plasma cortisol response to the test. Neither follicular phase women nor luteal phase women produced a cortisol response that reached statistical thresholds. But look at this. Luteal phase women showed the strongest subjective emotional responses. They reported elevated anger and hostility scores that persisted for 20 minutes after the test ended, higher than both men and follicular women. They also produced the largest allopregnanolone responses to stress, along with greater heart rate reactivity. The body was absolutely responding, just not through cortisol. Follicular phase women showed blunted noradrenaline responses. Luteal phase women showed amplified emotional and cardiovascular activation. The menstrual cycle was modulating, which channels the stress response flowed through, like water finding different routes down the same hillside depending on the season. So why do women show a dampened cortisol response? One answer lies in the endogenous opioid system. Beta endorphins, your body's own opioid molecules, maintain a constant inhibitory tone over the HPA axis. They act on mu-opioid receptors in the hypothalamus to suppress CRH release, essentially holding the stress axis on a leash at all times. When you give someone naltrexone, a drug that blocks opioid receptors, you remove that leash. And here's what happens. A placebo-controlled study found a clear sex difference. When the opioid inhibition was pharmacologically removed, women showed a heightened ACTH and cortisol response compared to placebo. Men showed no significant change. Consider what this means. Women maintain stronger opioid inhibition of the HPA axis at baseline. Their stress axis is being held in check more firmly, more actively. When you remove that restraint with a drug, the unmasked response is actually larger. The system underneath isn't weaker. It's being controlled more tightly. Estradiol increases mu receptor density and circulating beta endorphin levels. So during high estrogen phases of the menstrual cycle, there's more opioid activity at the receptors, producing stronger basal inhibition. The system isn't less capable. It's calibrated to a different set point, a different strategy for managing the same biological challenge. And there's one more finding here that connects beautifully to the aging story we'll tell next. A study analyzed paired 24-hour ACTH and cortisol time series from 48 healthy adults, ages 18 to 77. They found that sex determines the age trajectory of HPA axis function, and it does so in opposite directions. Total daily cortisol secretion fell with age in men, but it rose with age in women. ACTH efficacy, measuring how responsive the adrenal glands are to the pituitary signal, decreased with age in men, but increased with age in women. The male stress axis grows quieter over the decades. The female stress axis grows louder. Same architecture, opposite trajectories, likely shaped by the gradual decline of testosterone on one side and the withdrawal of estrogen after menopause on the other. Let that sit for a moment. Your stress system is changing direction, slowly, across the whole span of your life. And the direction it moves depends on which hormonal environment it's been bathed in. Speaking of hormonal decline, let's follow what happens when the body's sex hormones begin to recede, because the way they recede matters enormously. For women, this transition has a name everyone knows: menopause, and it doesn't arrive as a slow fade. It arrives as something closer to a cliff. Over a period of roughly 2 to 5 years surrounding the final menstrual period, estrogen levels drop rapidly. Not gently, not gradually, rapidly. And one of the most clearly measured consequences is what happens to bone. The study of women's health across the nation, known as SWAN, is one of the most thorough long-term studies of the menopause transition ever conducted. It tracked thousands of women across many years, measuring bone mineral density at regular intervals. The pattern it revealed is now considered canonical in the field. During the premenopausal and early perimenopausal years, there's little or no change in bone density. The skeleton is stable. Estrogen is maintaining the balance between bone formation and bone resorption, the constant process of building and dismantling that keeps every bone in your body alive and structurally sound. Then in the late perimenopause, roughly 1 to 2 years before the final menstrual period, bone loss speeds up. At the lumbar spine, it reaches 1.8% per year. At the total hip, about 1% per year. After the final menstrual period, the rate climbs higher. At the spine, 2.3% per year. At the hip, 1.4% per year. If sustained for 5 years, those rates reduce the average woman's spinal bone density by 7 to 10% and hip density by 5 to 7%. That corresponds to a 50 to 100% increase in fracture risk. A quiet number with loud consequences. There's another measure called trabecular bone score, or TBS, which captures aspects of bone quality beyond just density. TBS remains stable before menopause. Then it drops by 1.16% annually, beginning about a year and a half before the final menstrual period. In the postmenopausal years, TBS continues declining at 0.89% annually. Across a 10-year window bracketing the final menstrual period, the cumulative decline is 6.3%. Over a lifetime, women lose approximately 50% of their trabecular bone, the spongy interior scaffolding, and about 30% of their cortical bone, the dense outer shell. Roughly half of that total loss occurs in the first 10 years after menopause. A cliff, not a slope. For men, the picture is different. There's no equivalent hormonal cliff. Testosterone declines approximately 1% per year after age 40. No abrupt event. No sudden withdrawal. Just a gradual, steady reduction that stretches across decades. The corresponding bone loss follows the same gentle pattern. At the hip, longitudinal studies show men losing 0.3 to 0.5% per year. At the femoral neck, data from the Framingham study puts it at 0.38% per year. Overall, beginning in the 6th decade, men lose roughly 0.5 to 1% per year. Cortical bone in men declines about 3% per decade. Trabecular bone, 6 to 10% per decade. Over the full adult male lifespan, lumbar spine trabecular density declines by approximately 46%. That's not far from the 50% seen in women, but the trajectory is spread over many more decades. Same destination, very different road. The comparison at the spine is stark. Postmenopausal women lose bone at 4 to 8 times the rate of aging men at the hip. At age 50 and above, osteoporosis prevalence is 19.6% in women, but only 4.4% in men. The lifetime fracture risk after 50 is 1 in 3 for women, 1 in 5 for men. The fundamental difference comes down to the nature of the hormonal change. A cliff versus a slope. and the particular sensitivity of trabecular bone to estrogen withdrawal. Trabecular bone, with its high surface area and rapid turnover rate, responds intensely when the hormone that had been keeping its remodeling in balance disappears within a few years. The mechanism is specific. Estrogen normally restrains osteoclast activity, the cells whose job it is to dismantle bone. When estrogen is withdrawn, osteoclasts become more active, more numerous. Bone resorption outpaces bone formation. The skeleton begins to thin from the inside. In men, the slower decline of testosterone and its partial conversion to estradiol through aromatase, which continues to provide some estrogenic protection to bone, produces a more moderate imbalance. Real loss, but accumulating across decades rather than concentrating in one 5 to 10 year window. This brings us to a broader question about how aging unfolds. Is it a steady, linear process, a smooth decline from peak to end, or does it happen in waves with certain moments where the body changes faster than others? Dr. Tony Wiscore's laboratory at Stanford has been studying this question using a method that would have been impossible a generation ago, measuring thousands of proteins in human blood simultaneously. In a 2019 study published in Nature Medicine, his team measured 2,925 plasma proteins in 4,263 individuals aged 18 to 95. They used a technology called SomaScan, which employs aptamers, small molecules that bind to specific proteins, to quantify the protein composition of blood with high precision. You give a blood sample. The machine reads the concentrations of nearly 3,000 different proteins at once. It's like getting a molecular fingerprint of your entire body's current state. Their central finding overturned the assumption that aging is a gradual, continuous process. Instead, the team identified 3 distinct waves of synchronized proteomic change, not a smooth decline. Waves, crests, periods when the number of proteins undergoing dramatic shifts in concentration peaks simultaneously. The first wave appeared around age 34, the second around age 60, the third around age 78. Using just 373 of these proteins, the researchers could predict an individual's chronological age with a correlation of 0.93 to 0.97 across different groups. Even a reduced set of 9 proteins produced reasonable age estimates. Each wave reflected different biological processes, not the same decline repeating, different ones. At age 34, the dominant signature was a downregulation of structural pathways, extracellular matrix proteins declining, the scaffolding of the body beginning to thin. At age 60, the signature shifted to hormonal activity, binding functions, and blood pathway changes. Axon guidance proteins and epigenetic signaling proteins began exponential rises. At age 78, bone morphogenetic protein signaling emerged alongside continued blood pathway shifts. Proteins like CHRDL1 and MMP12 showed exponential increases. The overlap between waves was limited. Each was largely composed of different molecular actors, different proteins carrying different messages. And proteins that changed most dramatically in old age were often invisible to standard statistical models, which were biased toward detecting the subtler, more continuous changes that occurred at younger ages. The 60-year wave is the one most relevant to what we've been discussing tonight. its dominant proteomic signature, hormonal activity and binding functions. This is consistent with the menopausal and andropausal transitions and their downstream effects on blood composition. The hormonal shifts we've been describing aren't just local events in the ovaries or testes. They propagate through the entire body, altering the protein makeup of the blood, which in turn reflects changes in organs, tissues, and cellular processes across the whole system. A 2024 validation study using multiple types of biological data, including transcriptomics, metabolomics, cytokines, and microbiome composition, from different groups of people, confirmed similar nonlinear aging waves at approximately ages 40 to 45 and 60 to 65. The waves appeared across all data types and all cohorts. They're not artifacts of one measurement technology. They're built into how human biology ages. Wiscore's team then pushed the approach further. They developed methods to measure organ-specific biological ages from plasma proteins. By tracing approximately 15% of circulating proteins to single-organ origins, they discovered that nearly 20% of the population shows strongly accelerated aging in one organ. About 1.7% are what the researchers called multi-organ agers, people whose multiple organ systems are running on an accelerated clock. Accelerated organ aging carries real consequences: 20 to 50% higher mortality risk, and organ-specific diseases relate to faster aging of those particular organs. Individuals with accelerated heart aging had a 250% increased risk of heart failure. In a 2025 follow-up published in Nature Medicine, the brain emerged as what the researchers described as the gatekeeper of longevity. Having an extremely aged brain, as measured by the protein signature in blood, increased mortality risk by 182% over approximately 15 years. Having an extremely youthful brain reduced mortality risk by 40%. And here's the detail that connects this proteomics research back to the hormonal story running through our entire night. Of the 1,379 proteins that changed with age, 895, nearly two-thirds, were more predictive for one sex than the other. The aging process at the molecular level operates differently in men and women. The same proteins, the same blood, but different aging trajectories, shaped by the different hormonal environments those proteins have been immersed in for decades. The most recent preprint from Wiscore's laboratory, appearing in February 2026, extended this framework to more than 40 distinct cell types using over 7,000 plasma proteins measured across 60,000 individuals, making it the largest human plasma proteomics aging study to date. They found that 20 to 25% of people exhibit accelerated aging in a single cell type, while 1 to 3% show it across 10 or more. One finding in particular stood out. The APOE gene, the strongest genetic risk factor for Alzheimer's disease, showed what the researchers call antagonistic effects depending on the variant you carry. APOE4 carriers had older astrocytes but younger macrophages. APOE2 carriers showed the inverse. And for Alzheimer's specifically, extreme astrocyte aging conferred risk comparable to carrying the APOE4 allele itself. But APOE4 carriers who maintained youthful astrocytes showed reduced risk of developing Alzheimer's. It's not just the gene, it's the cell type housing it and how quickly that cell type is aging. Let's step back now and look at the broader pattern we've been tracing. We've followed hormones through the body tonight, from the hypothalamus to the muscles to the brain, through the stress axis, into the skeleton, out to the proteome of the blood. We've watched them ignite puberty, cycle through monthly rhythms, respond to social pressure, and withdraw across the decades of aging. And across all of this, one thread keeps reappearing. Male and female biology runs on the same cellular machinery. The same muscle protein synthesis pathways, ACT, mTOR, P70S6K, identical response rates, identical signaling, identical fiber type starting distributions. The same serotonergic and dopaminergic circuits, same TPH2 gene, same estrogen receptor beta at the same promoter site, same SERT transporter clearing serotonin from the synapse, same GABAA receptors binding allopregnanolone, the same HPA axis architecture, same CRH, same ACTH, same cortisol, same opioid receptors providing tonic inhibition. The same protomic waves washing through the blood at ages 34, 60, and 78, though two-thirds of the changing proteins are more informative for one sex than the other. The same trabecular and cortical bone structures, maintained by the same osteoblasts and osteoclasts, diverging only when estrogen withdraws rapidly rather than gradually. All of these differences, everyone, trace back to molecules measured in nanograms per deciliter and picomoles per liter. Concentrations so small they're invisible. Quantities you couldn't see, couldn't feel, couldn't hold. Shaping the skeleton, the brain, the stress response, the pace of aging over an entire lifetime. There's one more hormone we should spend time with before the night grows late. Dopamine. It's often called the pleasure chemical or the reward chemical, and those labels aren't exactly wrong, but they miss something important. Dopamine is better understood as a motivational signal, a signal of pursuit, of wanting, of moving towards something, not necessarily of enjoying it once you arrive. Dopamine has been described as a universal generic currency in the brain for pursuing, not unique to any particular desire, not specific to food or love or achievement or rest. It's a shared signal that drives the brain toward goals of all kinds. The object doesn't matter. The dopamine system is the engine underneath. And this engine is modulated by hormones. Estrogen increases dopamine synthesis and decreases its degradation, particularly in the prefrontal cortex. Testosterone modulates dopamine pathways too, though through different receptor interactions and in different brain regions. Both sex hormones adjust the autonomic nervous system, the background regulation of heart rate, breathing, arousal, which in turn influences how dopamine signals are interpreted and acted upon. But here's the central understanding, and it applies to everything we've covered tonight. Hormones do not dictate behavior. They don't command, they don't force. What they do is adjust the probability of certain responses. They lower thresholds or raise them. They make some reactions more accessible and others less likely. A surge of cortisol doesn't force you to panic. It makes the panic response easier to trigger, closer to the surface. A rise in estradiol doesn't create happiness. It increases serotonin availability, which adjusts the brain's emotional baseline in a direction that, for most people, registers as slightly more buoyant. Hormones are suggestions, whispered continuously. You can override them. You can learn to work with them. You can recognize their influence without being controlled by it. The autonomic nervous system, which hormones are constantly adjusting, can also be self-regulated through deliberate breathing, through focused attention, through patterns of response you build over time. This isn't to minimize hormones. Their influence is real, measurable, and unceasing. But the relationship between a hormone and a behavior is probabilistic, not deterministic. A hormone sets the stage. It doesn't write the script. And the stage changes, moment by moment, day by day, month by month, decade by decade. The hormonal environment you inhabit right now isn't the one you inhabited 10 years ago, and it won't match the one you'll inhabit 10 years from now. Your body is always recalibrating, always adjusting the blend. Despite testosterone levels that differ by 10 to 20 times between men and women, the cellular machinery of muscle responds almost identically to the same stimulus. Same protein synthesis signals, same fiber type adaptations, same growth pathways, same response rates. And yet a single methyl group, smaller than a molecule of water placed on DNA during puberty, can alter gene expression for decades, shaping how the body builds bone, how it manages stress, how it processes emotion, how quickly its cells age. The wonder isn't in the differences between male and female. It's in how much is shared. The same quiet biochemistry, tuned to slightly different frequencies, by molecules measured in nanograms and picomoles, running beneath everything, always running. Consider what we've traveled through tonight. We began with the shared blueprint, the same hormones, the same receptors, the same hypothalamic pituitary control system present in every human body, regardless of sex. We followed that blueprint into puberty, where a methyl group lifted from a gene in the hypothalamus set off a cascade that diverged male and female development, not by creating different machinery, but by tuning the same machinery to different settings. We watched epigenetic marks laid down during those years persist across decades, shaping how the genome reads itself long after the visible changes of puberty have finished. We traced the monthly rhythm of estrogen and progesterone into the brain, watching these fat-soluble molecules cross the blood-brain barrier and adjust serotonin synthesis, serotonin clearance, GABA receptor sensitivity, and dopamine activity in regions governing mood, reward, and decision-making. We noted that this rhythm is not a defect. It's the body cycling through different neurochemical states as part of a reproductive process hundreds of thousands of years in the making. We examined the assumption that testosterone drives muscle protein synthesis and found it unsupported by careful measurement. Same signaling proteins, same growth response, same fiber type distributions, same adaptations. The difference lies in the starting mass, set during puberty, not in how cells respond to effort. We stood inside the Trier social stress test and watched men produce 58% higher peak cortisol, while women reported similar subjective distress. Not a weaker female response, a differently calibrated one, maintained by stronger opioid inhibition at baseline, modulated by menstrual cycle phase, and changing in opposite directions across the lifespan. We followed bones through menopause and andropause, the cliff and the slope, the same skeletal structures responding to two very different patterns of hormonal withdrawal, producing loss rates that differ by a factor of 4 to 8 at the spine. And we looked at the proteomics of aging, where 3 waves of molecular change wash through the body at ages 34, 60, and 78, with the 60-year wave dominated by hormonal signatures and nearly 2/3 of aging-related proteins showing sex-specific patterns. The body is not a machine switched on by hormones. It's a conversation between molecules, a conversation that lasts an entire lifetime. Right now, as you rest, cortisol is quietly lowering. The HPA axis is stepping down its activity, following the circadian rhythm that peaks in the early morning hours and recedes through the evening. Melatonin is rising, released by the pineal gland in response to the dimming of light reaching your eyes. Your body is shifting into its own nightly hormonal rhythm. This happens whether you notice it or not. The same hypothalamus that directed the changes of puberty, that orchestrates the monthly cycle, that calibrates the stress response, that same small structure is now gently steering your body toward rest. Same pituitary, same chemical language of hormones drifting through the blood, same cells listening. And as you settle deeper into the night, growth hormone begins its own quiet work. During the deepest stages of rest, the pituitary releases growth hormone in pulses, not a constant flow, pulses, slow rises and falls, synchronized with the architecture of your body's nightly repair cycle. These pulses reach muscle cells, signaling them to mend. They reach bone cells, supporting the slow maintenance of the skeleton. They reach the brain, contributing to the consolidation of memory, the process by which the experiences of the day are woven into the longer fabric of what you'll remember tomorrow, and next year, and decades from now. This has been happening every night since you were born. Growth hormone pulses, rising and falling, doing their quiet work of maintenance and repair while the conscious mind is absent. You don't direct it. You don't control it. It just happens, night after night, year after year. The hormones continue. They continue when you're not thinking about them. They continue when you're not measuring them. They flowed through you during every conversation you've ever had, every effort you've ever made, every moment of stillness. They shaped your bones when you were too young to know what bones were. They adjusted your brain's chemistry during every emotion you've ever felt. And they are adjusting it now in this moment as the night deepens around you. Estrogen, testosterone, cortisol, melatonin, growth hormone, dopamine, serotonin, allopregnanolone, kisspeptin. Molecules with names that sound clinical in the daylight, but do their work in silence. The conversation between them is older than language, older than thought, older than every human invention, every culture, every story ever told around a fire or whispered in a dark room. It was running in your ancestors. It runs in you tonight, and it will continue adjusting and recalibrating through whatever years lie ahead. The cells listen. The molecules drift. The body adjusts. And somewhere within this quiet chemistry, within this slow, continuous exchange of signals too small to see and too constant to notice, the night settles a little deeper. The hormones continue their work. The body rests, and the conversation goes on. Sweet dreams, and we will see you next time on Bedtime Science.